Okay, listen carefully. What I'm going to cover in Chapter 12, if you were taking care of your responsibilities, especially for labs 10, 11, and 12, you should notice a lot of this, okay, feeds into those three labs, or from the reverse perspective, these last three labs you completed feed into this chapter. So at the very least, hopefully you'll recognize some of the information we're going to go over, and if everything goes well, it'll start to make a little bit better sense, okay? All right, so this chapter, it says Chapter 12, DNA Technology. The term they use now is biotechnology. So if you hear somebody talking about biotechnology, it's just basically it involves a study of life. In my day, we didn't say biotechnology. If you were working with DNA, from any perspective, we called it recombinant DNA technology. So it's more specifically, that's what we're going to be looking at here. Okay. If you recall, I said all living organisms have their genetic information stored in the form of DNA. And because of that, scientists realize that not only do parents pass on traits to offspring through this compound DNA, but they realized they could manipulate organisms, creating GMOs, genetically modified organisms, either at the unicellular level or at the multicellular level, simply by, in one species, locating, say, a gene of interest, extracting and isolating that gene, and inserting it into a completely different species, thus transforming that different species' genotype and therefore its phenotype. And often this is done to enhance the traits, okay, of the species that's receiving the foreign gene. So here it's showing you, because all organisms use the same genetic code, meaning it's stored in DNA, scientists can make a plant glow like a firefly. So in this case, they didn't use GFP. In this case, what they did is they cloned a gene that codes for an enzyme called luciferase. Okay, and if the plant produces the necessary reactants or substrates, that enzyme luciferase, okay, will create molecules that fluoresce green. Well, the way it worked early on, and this was developed at least by the early 1970s, or early to mid 1970s, it was a process they called DNA cloning. Okay, and cloning just means making copies. In this case, if it's DNA cloning, we're talking about making many copies of the same DNA molecule. And what they would do is initially when they would isolate their DNA of interest, because sometimes the DNA they isolated, they didn't know what it was, they just knew it was DNA. So they wanted to isolate that DNA, make as many copies of it, identical copies of it as they could, so they had a larger quantity to study, to be more easily study. Sometimes, however, they knew what DNA they were extracting and isolating. It could be a gene, or it could be something like a regulatory sequence, a promoter, a terminator, an enhancer, or a silencer DNA sequence that they wanted to study more in depthly. But first they needed to isolate it and make many identical copies of it. So we had DNA cloning. But initially what they found out is it wasn't enough to identify an organism that could perform the cloning process and identify the DNA of interest that they wanted to clone. They came to find out that the DNA of interest they wanted to study, therefore they wanted to first clone it to make enough copies of it to study. They couldn't just insert it into an organism that would make copies of it. What they found is they had to create what some scientists call plasmids. Some scientists call vectors. I worked in a lab where we worked with a lot of DNA. And I used to ask those that were training me, why do you say plasmid and vector back and forth? And they told me they didn't know. So as I became more familiar with working with DNA, to me, as I'll talk about, a plasmid is bacterial in nature. There are small circular pieces of DNA that are not part of the main DNA that forms a bacterial chromosome. Small circular pieces of DNA that you will find in a bacterium. Okay. A vector, it is also a circular piece of DNA. It is double-stranded DNA, just like a bacterial plasmid. However, this does not naturally occur or is not naturally produced in living organisms. It's produced in vitro in a tube, and it consists of, okay, DNA from different organisms, such as the vector we used in the lab. I'm going to use this same vector for the lecture. Okay, so hopefully seeing it twice makes a little sense. A piglo vector. I use the term vector, and the way I define a vector is a vehicle or carrier used to get your DNA of interest, okay, into a cell. Now that DNA of interest, okay, there are different reasons to do it as we'll go over, but one of the reasons why you would want to get your DNA of interest using a vector into a cell is simply to get that cell to make copies of it for you. So this P-glow vector, it has information from jellyfish, but it also has information from the bacterium E. coli. So this is what we call a vector. A vector, if you want to call it, it's synthetic. It's made in a tube, whereas a plasmid is bacterial in nature. So that's what I'm going to talk about in this chapter. But what they found is initially early on, in order to make these copies of DNA, in order to perform DNA cloning, they had to insert their DNA of interest, such as this jellyfish gene, Gfp gene, into a cloning vector. Okay, then they would insert it into bacterial cells, which they found worked great, such as E. coli. And every time the E. coli divided, well, the cloning vector had an origin of replication. So every time the E. coli divided, it would not only copy its own or replicate its own DNA, it would replicate the cloning vector because it would notice the ori. Then what we would do is after 24 to 48 hours, okay, we would take those bacterial cells that all had the same cloning vector in them, okay, and we would extract and isolate the DNA from them. And we would isolate it and we would store it in a tube. So here it's showing you clone genes were available in a test tube, but such as a clone gene, such as GFP gene, but they're showing you the circular piece to show you it's part of a larger piece of DNA called a vector. And this is what we used to do in the lab. Now times have changed. You don't have to use bacterial cells, okay? But as I said, the cloning vectors that we would use to clone our DNA of interest, to make copies of our DNA of interest, had to have an origin of replication. Now, what I didn't mention is the reason why the cloning vector has to be circular in nature was because we were using bacteria to perform the actual cloning process, making copies of the vector, therefore making copies of our DNA of interest, which was part of the vector. Because bacteria such as E. coli or prokaryotes more in general, they don't have linear DNA like you find in eukaryotic cells. They have circular DNA. So that's a big difference in terms of prokaryotic versus eukaryotic DNA. Just based on appearance, simple appearance, prokaryotic DNA, such as bacterium E. coli, it's always double-stranded helix, but it's circular. Whereas with eukaryotic DNA, it's linear in nature. It has open ends. But when you put the cloning vector or when you insert it into a bacterium such as E. coli, when the bacterium copies its own DNA, it also replicates your cloning vector. So that's the purpose for this origin. It's actually helping make copies of our DNA of interest when it clones the entire vector. But as I said, a plasmid is bacterial in nature. So this is showing you the cytoplasm of a bacterium. Okay, the remnant here just means it's you can just think of it as proteins with DNA, and then you can see some of the DNA extending off of it. They show you, they don't say DNA, but remember, if you see the term chromosome, chromatin, or sister chromatids, we're talking about DNA. So instead of calling it bacterial DNA, they call it a bacterial chromosome. What you can't see here is that technically this is circular in nature. It's just coiled up real tight, bunched together real tight in the form of a chromosome. But notice something over here. You can see a little separation from the bacterial chromosome and this little piece right here. This is also a circular piece of DNA. If you go over here, you can see a little separation between the bacterial chromosome and this little piece here. Well, they're showing you these two little round circular pieces are plasmids. Bacterial plasmids, okay, are DNA, and they are circular pieces of DNA, although you can see it's all folded up. It is a circular piece of DNA, which is why they show you this here, trying to show you. But notice the plasmid, the bacterial plasmid, is not part of the main DNA. It's not part of the main bacterial chromosome. However, bacterial plasmids were found to carry genetic information, such as genetic information that would allow the bacterium to be antibiotic resistance. Okay, so if you recall in lab, that P-glow vector had a gene called the BLA gene. That BLA gene was discovered in a bacterial plasmid in one of these little pieces. Now, the entire plasmid did not form, the BLA gene did not make up the entire plasmid. It was just part of the plasmid, okay, contained that BLA gene. And what they came to find out is that BLA gene coded for an enzyme that would destroy the antibiotic ampicillin. So for the cloning vectors, such as pGlo, what they ended up doing is isolating that bacterial plasmid that had the BLA gene, and then they extracted and cut out or isolated the BLA gene and used it to create a cloning vector. Also for cloning vectors though, what they often like to do is add another type of gene besides an antibiotic resistant gene. They like to add what you call a reporter gene. The way we define a reporter gene is a reporter gene codes for a protein that is easy to detect. So again, a reporter gene codes for a protein that is easy to detect. You can't see the DNA molecules. Okay, even when you create your cloning vector, you can't see it. The closest you can come is see it in a DNA gel electrophoresis, but even then you can't see the actual structure of it. Likewise, when you insert this cloning vector with your gene of interest into bacterial cells or even eukaryotic cells, you don't know if you're successful. Thus, we use the BLA gene for antibiotic resistance to ensure that only transformed cells can grow in the presence of an antibiotic, but also to further ensure that the vector is that we successfully transformed a cell and inserted the cloning vector into a bacterial cell. We also have a reporter gene. A reporter gene is any gene that codes for a protein that will be detected. So down here, that Gfp gene, which you see here, okay, that Gfp gene is also considered a reporter gene. Why? Because if you think about it, had we been able to do the lab 12 in person, the genetic engineering lab, how would you have known if you had successfully transformed your E. coli cells? By simply taking out the petri dish that contained the bacterial cells you transformed, shining a UV light on them, and if you had successfully inserted your pGlo vector into those cells, well, those cells would have been expressing green fluorescent protein, which would have simply fluoresced green, therefore easy to detect. So that's what a reporter gene is. A reporter gene is a gene such as the Gfp gene that codes for a protein that can be easily measured or detect. In this case, Gfp simply fluoresces green. This is why Gfp is so popular. It made it so much easier to determine if your experiment was successful in terms of I was able to transform this organism. I was successful in inserting my vector. So I'm going to go over this vector. So you saw it in lab, but you also see it on the test here. And I started doing this a few years ago. I actually used to go over a different cloning vector that was real popular, but then I realized that I needed to double up on this vector between lab and between lecture, and hopefully it would help with grades on both sides by having some repetition. Sorry, excuse me. So the pGlo vector, this is going to be repetition, so hopefully this will make sense. The four key parts that make up the pGlo cloning vector. Hold on, everybody, real quick. All right, sorry, everybody. So if you look at the pGlo cloning vector here, okay, it has four key parts: origin of replication and three genes: BLA gene, Gfp gene, ARIC gene. Okay, and we'll go through these one at a time as we go. We already talked about it. The purpose of the ORI is origin of replication, is to allow the bacterium that you have transformed, successfully inserted your cloning vector into, to allow that bacterium to not only replicate its own DNA when it divides from one cell into two, but also to replicate your cloning vector that contains your gene of interest. Therefore, when the vector is cloned and passed on to two cells now, each of these vectors consists of your DNA of interest, so your DNA is also cloned. And if you recall, I had said this last class, each individual bacterial cell that you successfully transform by successfully inserting your pGlo vector into, each of those single transformed cells will eventually divide into a large number of identical cells or clones. So if you look here, the Petri dish, had we been able to meet in person, after 48 hours, we would have removed your plates from the 37-degree incubator, and you would have seen these little white dots. Each of these white dots is not a single bacterial cell. Each of these little white dots is a group of genetically identical bacterial cells. But instead of saying a group of genetically identical bacterial cells, We refer to each of these white dots as a bacterial colony. Okay, now for the 3 genes that make up the cloning vector, the BLA gene is referred to as an antibiotic resistance gene. Why? It codes for an enzyme called beta-lactamase. So when you successfully insert this cloning vector into a bacterial cells, the ones highlighted in pink here, okay, these cells recognize this genetic information in the form of DNA, and they start to express this BLA gene, resulting in the production of protein called beta-lactamase. So remember, any gene that is carried in this vector, if the gene is working, if it is actually not only changing the genotype but the phenotype of the transformed bacterial cell, then that means the gene is undergoing gene expression, transcription, translation, resulting in the production of a protein enzyme called beta-lactamase. And they found that beta-lactamase destroys an antibiotic known as ampicillin. So whenever you successfully transform a bacterial cell by inserting the pGLO vector into it, now what you have passed on is its ability to destroy the antibiotic ampicillin. Therefore, it's resistant to it. Therefore, it will not die in the presence of that antibiotic. However, any cell that you did not successfully transform, did not successfully get the vector into, it does not have this BLA gene. Therefore, it does not produce this protein enzyme. Therefore, it cannot destroy ampicillin. And as you can see, going from here to here, these cells are killed off by the antibiotic, and the only surviving cells are the ones that, took up the vector with the antibiotic resistance gene. They produce beta-lactamase, which destroys the antibiotic in the growth medium here, and they not only survive, but they start to divide using the origin of replication here, producing a colony or a bacterial colony of identical or genetically identical bacterial cells. Well, we call this selective growth, and that's the main purpose for the BLA gene. It's not just to convey or pass on the trait for antibiotic resistance to bacteria that originally were not resistant to the antibiotic ampicillin, but it's to allow us to perform selective growth. This ensures only our bacteria that contain the cloning vector, the recombinant DNA, this ensures those are the only cells that are growing, and therefore we don't have to go through each cell screening it, trying to figure out, did you take up the vector? Because that's how they used to do it. They would analyze tens of thousands of bacterial cells looking for the one that they successfully transformed, the one they successfully got the vector in. But by coming up with this antibiotic resistance gene and using it for selective growth, this helped cut down the screening time. Now they know the only cells surviving are transformed. They were selected for. And then again, the GFP gene, as I said, it's actually a reporter gene. Okay. The GFP gene codes for a protein called green fluorescent protein that when you expose it to UV light, it causes the organism to fluoresce green. The protein itself fluoresces green. Thus, it's easy to detect, and in this case, it makes it even easier to determine. Even without the BLA gene, it would still make it easier to determine, did you successfully transform your bacterial cell or did you successfully transform your organism? Because it simply fluoresces green upon exposure to UV light. And that's the definition for a reporter gene, as I said before. You can see here, they're showing you E. coli that were not transformed using this vector, and E. coli that were transformed using this vector. And you can see under UV light, they simply fluoresce green. So technically speaking, if you have a reporter gene that works as well as a GFP gene, you could actually get by without using the BLA gene, the antibiotic resistance gene, because this one makes it even easier to tell if you're successful in transformation of your bacterium. Then there's the ARSC gene. I have said this before in the lab overview on Tuesday. What geneticists or scientists in general are trying to do is they're not only trying to create GMOs by transforming their genotype and therefore their phenotype, but when they insert those foreign genes into an organism that normally doesn't have that genetic information, they want to be able to, if you want to call it exogenously or externally, still be able to control when that gene works in the organism. Okay, so this is the case for the ARSC gene. Again, I said this before, the ARSC gene codes for a protein that is a regulatory protein, but more specifically, it's a transcription factor, specifically an activator. So if you compare it to your notes I gave you in chapter 11, the ARC gene would code for a protein called ARC, and if you were to take ARC and place it in your chapter 11 notes, you would place it under transcription factors called activators. That's where this would go. Now, it's not enough for this gene to be expressed in a transformed bacteria cell, resulting in the production of ARC protein. The protein won't work unless you feed the bacterium a sugar called arabinose. Okay. Well, this ARC gene was actually discovered in E. coli, just like the blonde gene. And it was through the studies of E. coli that they found that whenever E. coli is exposed to arabinose, it's a source of nutrients. Okay. But it also activates this protein, helping E. coli metabolize it. So they just manipulated it and put it into a cloning vector to where now it works in a different manner. When you get this cloning vector successfully into a bacterium, now what happens is if you feed the bacterium arabinose, it'll take up this sugar. Some of this sugar will bind to this ARC transcription factor or activator. This will activate the ARC, promoting it or causing it to help RNA polymerase locate the Gfp gene and activate and express the Gfp gene. But arabinose has to be present. So there's more to the vector than what I talk about, but the bottom line is the only way the Gfp gene will be expressed in a transformed bacterium, okay, is if you feed the bacterium arabinose so that the protein produced by this gene, so that protein will become active and come over here and help RNA polymerase find this one. So we call it a molecular control switch. Okay. Now the purpose for this is because these molecular switches is because you may have heard a lot of talk about gene therapy and CRISPR technology, starting off with focusing on those with genetic disorders and eventually moving over to, say, type 1 diabetics and things of that nature. But they know that it's not enough sometimes to just repair, okay, the mutated genetic information that's not working properly, and therefore the organism's phenotype, you can see the negative impact it has on the phenotype. They realize it's not always enough, okay, to correct the mutation at the level of DNA. They also have to be able to control when that, corrected gene or fixed gene is expressed. It can't just be allowed to just work at any time. Just like in us. Insulin gene, okay, there are pancreas cells that express the insulin gene producing insulin, but we don't want insulin produced 24 hours a day, 7 days a week. We primarily only want insulin produced when we eat or just after we eat, okay? So this is what they're getting towards. Now, what I'm going to go over now are enzymes that were discovered in bacteria, and it was these enzymes that allowed for creating cloning vectors such as the pGLO vector. Because if you recall what I said, okay, I said a vector is a vehicle or carrier used to get your DNA of interest into a cell. However, a vector is made up of DNA from different organisms. Well, how were scientists able to create the vectors? It was through the discovery of enzymes called restriction enzymes. Scientists discovered that even though viruses such as bacteriophages, okay, not only infect bacteria, but obviously transform them into viral reproducing cells, host cells, and then eventually killing the cell, they found out that bacteria, just like us, they have their own immune system, everybody. They can protect themselves from a viral infection. There are at least a couple of ways that they do it. One of them is through the expression of proteins called restriction enzymes. Restriction enzymes are digestive enzymes that cut up or digest DNA. Okay. What they found, though, is in bacterial cells. For the bacterial cell itself, the bacterial DNA has what we call a methyl group attached to it. These are functional groups made up of carbon and 3 hydrogen. And what they found is bacteria, whenever they replicate their DNA, when they're dividing, they'll attach methyl groups throughout their DNA. These methyl groups serve as markers or tags to let the enzymes they express here in green, to let their enzymes that the bacteria express know, don't cut up this DNA. It belongs to us. We're on the same side. But what they found about these restriction enzymes is that this ensures that the restriction enzymes only cut up or destroy DNA that, say, comes from a virus and not its own DNA or the DNA that's part of the same cell. So now what happens by the bacterial cell tagging its own DNA with methyl groups, now the bacterial cell's restriction enzymes that it's producing know, hey, look, now I can tell the difference between viral DNA and our own DNA. And whenever a bacteriophage is attempting to infect a bacterium and trying to transform it into a host, these restriction enzymes actually look at certain combinations of bases that they call restriction sites. So what scientists came to discover is that these enzymes, the way they recognize DNA is by reading the bases in the double-stranded DNA and recognizing certain combinations of bases. And when they see these certain combinations of bases that we call restriction sites, the restriction enzyme will bind to the restriction site that is part of the DNA and then simply cut it up. Well, this fascinated scientists. So eventually what they found out is these restriction enzymes are proteins. So eventually they backtracked and identified the genetic information that codes for these enzymes. Some of these restriction enzymes are what we call endonucleases. What they do is they cut DNA from within the double-stranded helix. So it doesn't matter if the DNA is linear, such as I showed you over here for the viral DNA. It doesn't matter if the DNA is linear or circular. If that DNA doesn't have methyl groups attached to it, the restriction enzymes called endonucleases will go within the DNA sequence, look at the base pairs, look for a certain combination of bases, and then the restriction enzyme will grab on to that certain combination of bases. Again, listen carefully. The protein is called a restriction enzyme. That combination of bases that it recognizes within the DNA molecule, that is the restriction site. So please remember that. If I mention restriction enzyme on the test, I'm talking about a protein that is an enzyme that cuts up DNA. If I mention restriction site, I'm referring to the base sequence in the DNA base sequence that the enzyme recognizes, binds to, cuts the DNA. So here where the DNA is cut, the DNA was cut at the DNA restriction site here. Well, to give you a couple of specific examples, two popular types of restriction enzymes isolated from bacteria are EcoR1 and small one. Now, I'm not going to give you the name for small one, but it's a long name. I'm going to use EcoR1 because it's easier. The way they name these restriction enzymes begins with the bacterium, the genus and species of the bacterium that they discovered the restriction enzyme in. EcoR1 was discovered in bacterium called E. coli. The R here represents the strain, E. coli strain R. The Roman numeral 1 here represents it was the first restriction enzyme discovered in E. coli. Okay. In the labs, we used to use EcoR1 and EcoR5, and the numbers just have to do with the order that they were discovered. Now, they're not showing you this, but imagine this EcoR1 looks more like this. It's a protein. In this figure, though, what they're showing and what they're highlighting is what I mean by restriction site. And I gave this to you on your notes, but listen carefully. On your notes, I gave you the restriction site based on the forward strand, because this is the strand that geneticists primarily use to identify the restriction site for restriction enzymes. And what they found for EcoR1, this restriction enzyme, it's not only expressed by E. coli to protect it from viral infections, but when this enzyme scans viral DNA, when it comes across this combination of bases, G, AA, TTC, along the forward strand, the enzyme will grab on to both strands and will cut the DNA, except this enzyme makes what I call a serrated cut. It doesn't make a straight cut through the double strands. It makes a cut here and it makes a cut over here, so that when it cuts the viral DNA, what ends up happening is we have what we call 5' overhangs, which in lay terms are known as sticky ends. Okay, and I'll explain why we call them sticky ends in a sec. But there are other bacterium, and I'm not even going to give you the name of this one. It's an ugly name, but again, it comes in the genus and species, and in this case, it was just not a strain, but just the first enzyme discovered. They call it small 1. This is a restriction enzyme, and it's an endonuclease. However, it recognizes a different restriction site. This restriction enzyme, when it comes across viral DNA, it'll scan the viral DNA, and it's looking for a restriction site, but the restriction site it recognizes, based on the forward strand, it recognizes a restriction site that reads CCC GGG. When it recognizes this restriction site, it will bind to both double strands, and it will make a clean cut right down the middle of the restriction site. This produces what we call blunt ends. Notice there's no single strand sticking out like we had with EchoR1. There's no overhangs. Therefore, there's no sticky ends. We call these blunt ends. Okay. The type of enzyme that we preferred to use in the lab when we did this was enzymes that created sticky ends. Okay, and the reason for that has to do with this. What we would do is we would perform a process that we called in lay terms cut and paste. Okay, we would use a restriction enzyme such as EchoR1 to cut out our DNA of interest and to cut open our vector. Now it says plasmid, but just assume it's vector. I forgot to change this, okay? Imagine this is your vector, not your plasmid. We would use a restriction enzyme to cut out our DNA of interest and to cut open our vector, open it up. If we could, we would use the same restriction enzyme and a restriction enzyme to produce sticky ends, such as EchoR1. So what would happen would be when we cut out our DNA of interest, it would have these sticky ends. When we would use the same enzyme to cut open our vector, cut it one time to open it up, because it's circular, we want to open it up. And even though it's not linear, you got to imagine we can unfold it and it's linear. But by using the same enzyme, such as EchoR1, to isolate our DNA of interest and cut open our vector, we end up with 5' overhangs, small single stretches. Okay. We end up, imagine this is a piece of the cloning vector. Imagine this is your DNA of interest. We end up with 5' overhangs that complement each other. This is the reason why we call these overhangs sticky ends. Because this strand, this single strand of TTAA, it will stick to the bases over on this DNA of interest, AATT. So this is why we call them sticky ends. They will stick to each other because they'll base pair with each other. Then what we do is we take an enzyme called DNA ligase, which is right here. We talked about this enzyme in DNA replication. We then take an enzyme called DNA ligase, and what it does is it creates the covalent bonds along both strands. Okay, to ensure now that we have a circular piece of DNA and both strands of the DNA have a sugar phosphate backbone that is not missing any covalent bonds. Once we create this, so this is why we call it cut. This is the cut part, and then using ligase is our paste part, pasting this in. Then we would go on and perform our transformation process. Okay, so what's the purpose now? What we're going to go over is the purpose of these different techniques. Well, the purpose of cloning vectors now, today, is to create GMOs that will produce human insulin, or to create GMOs, plant GMOs, that are resistant to insects, or so that they can serve as a better food source. So down here, what they're showing you is that by creating these cloning vectors, and then by using particular pieces of DNA to create that vector, we can end up transforming, say, bacterial cells with a cloning vector that has human genes that produce proteins that help prevent blood clots. Or we can insert a cloning vector into bacterium that allows them to produce an enzyme that helps them digest the carbon compounds in an oil spill. Or what we can do is we can transform not bacteria, but plant cells, embryonic plant cells. Okay, and we transform them with a cloning vector that contains a gene that codes for an endotoxin that kills insects, eventually creating GMO plants that are resistant to insects, and we don't have to treat them with pesticides or insecticides. So this was the purpose eventually of these cloning vectors. And it says here, animals, plants, and even bacteria can be genetically modified to produce human what? proteins. There's another technique, though, and this technique's been around for at least as long as cloning process, so at least from the 70s, maybe earlier. It's called DNA gel electrophoresis. And what DNA gel electrophoresis does is it basically allows you to analyze your DNA in terms of, do I have DNA to start with? And if you know information about your DNA in terms of the size of the DNA gel electrophoresis also helps you be able to analyze the size of that DNA. And sometimes that size can be related to your DNA of interest. So you confirm that you have the DNA you want based on the size. What scientists realized is that because the DNA nucleic acid monomers that form DNA nucleotides, if you recall, they're made up of 3 small molecules, a base, a sugar, and a phosphate group. That phosphate group has a what charge to it? A negative charge. Therefore, when DNA nucleotides are used by a cell to form double-stranded DNA, the overall molecule of double-stranded DNA is negatively charged. So scientists started to realize, you know what? We can actually take advantage of this charge. When someone discovered, and I don't know who discovered this, but someone discovered that seaweed produces a polysaccharide called agarose. And they found that when they isolate this polysaccharide and produce it in powdered form and then dissolve it in a buffer solution at a high temp and then pour that agarose solution into a container that allows it to creates a rectangular shape, that when this agarose solution cools, it solidifies into a porous gel. And what they found is taking advantage of the negative charge of DNA, taking advantage of this polysaccharide called agarose that can create a porous gel, and then taking advantage of just electricity. These 3 components can be put together and allow you to separate DNA based on size. Separate longer or larger fragments from shorter or smaller DNA fragments. Okay, so therefore what you would have done had we did it in a lab is you would have seen the gel itself, seen the shape of it. We would have put the gel in a buffer solution that helps with the electrical current passing through the gel. You would have loaded your DNA in solution on one side of the gel that you would have applied your negative charge from the cathode, because negative and negative repel each other. So that would have pushed your DNA fragments into the gel. From the other side of the gel, you would have had an anode which emits a positive charge that attracts a negative charge. So that would have pulled your DNA or attracted it. So you're pushing your DNA from one end through the negative charge, and you're attracting it or causing it to move through the gel towards the opposite charge or positive charge. But instead of saying push and pull, we say it causes DNA to migrate through the gel based on size. The smaller the length of the DNA fragment, the faster it moves. The longer the length of the DNA, the slower it moves. But this is a catch. Whenever we perform DNA gel electrophoresis, okay, we don't like to use circular DNA. If there is a circular piece of DNA, we try to use a restriction enzyme to cut it one time to linearize it, to make it more of a straight piece, because that allows us to better separate it based on size. So whenever you perform DNA gel electrophoresis, you're separating DNA fragments that are, if you want to call it, linear lengthwise. Purpose of gel electrophoresis: paternity testing, forensic science. You just did it in lab. You had 2 labs. So we didn't get to do it hands-on, but you had 2 labs. We used DNA electrophoresis for cancer gene detection. You used DNA gel electrophoresis for paternity testing. And the paternity testing involved DNA fingerprinting. So that's what you took advantage of. Now, PCR. Everybody, this technique called PCR was a very powerful invention, very powerful tool. When the idea and then the actual, if you want to call it, application of this idea occurred, it revolutionized biotechnology. Okay, so if you were to do your homework and dig back into the past in science in regards to the Human Genome Project and DNA fingerprinting being used for forensic science to convict criminals, you'll see these areas date back to the 90s, because this technique, PCR, really wasn't perfected till the 90s. But once it was perfected, scientists, including forensic scientists, came to realize this is a powerful tool. We can really use this to help us accomplish our goals. PCR stands for polymerase chain reaction. And what PCR is it's DNA replication in vitro. If you ever see the term in vitro, automatically tell yourself in a tube or in a test tube. Okay, so this is what PCR is. It's DNA replication in a test tube. The idea came about in the early 1980s, but the ability to actually apply it didn't come about till roughly the early 1990s. Okay, in order to perform this process PCR, you need to have your DNA of interest, which we call your template, but you also need that enzyme that we talked about in chapter 10, DNA polymerase. The problem was the person that came up with this idea for PCR, his name was Kerry Mullis. When he came up with the idea, the problem was the only known DNA polymerases, okay, had been discovered in bacteria or in us, in other mammals. The problem with DNA polymerase from bacteria and mammals is that DNA polymerase, it's a protein enzyme that works best at 37 degrees. And the problem with that is in order to perform PCR, you have to use 3 different temperatures, and the lowest temperature is 59 degrees. Therefore, the only known DNA polymerases that could be used for this in vitro process of PCR, those enzymes would denature. Therefore, DNA replication wouldn't occur. So roughly around the mid to late 1980s, scientists definitely discovered that there were organisms that survived at very high temps by volcanic vents, and they were aquatic in nature. They came to call them thermophiles. And when they analyzed these thermophiles, they came to find that these thermophiles, just like bacteria and just like mammalian cells, have produced DNA polymerase, and that the DNA polymerase in these thermophiles performs DNA replication, except the DNA polymerase, okay, that you found in thermophiles, it did not denature at high temps because thermophiles live at temperatures roughly 77 degrees Celsius up to 121 degrees Celsius. They not only survive, they reproduce, they thrive at these temperatures. So eventually scientists isolated the DNA polymerase produced by these thermophiles, and they were able to use it for PCR. Because DNA polymerase from thermophiles doesn't denature at high temps. The highest temp you use for PCR is 94 degrees Celsius, and they found out that thermophile DNA polymerase does not denature. So this was one of the huge discoveries that allowed for the application of this technique. The other, how do you say, if you want to call it discovery or invention that allowed for the application of PCR was this piece of equipment. This is known as a thermocycler. Okay. What you would do is you would take a little test tube called PCR tubes. You would add your DNA of interest. You would add the thermophile DNA polymerase and some other components to a small test tube. And then you would place the test tube up here in the top. Okay. He's removing the cover, but you would place it here in the yellow part on this instrument here. We have an instrument similar to this in our teaching lab. Once you put the PCR tubes there, you would close the lid. Then this thermocycler, okay, has a computer program in it that allows you to program the different temperatures you will need to perform PCR. The lowest temperature is 59 degrees, the highest temperature is 94 degrees Celsius. Okay, but that's fine because the enzyme in our PCR tubes, it's thermophile DNA polymerase. It doesn't denature at high temps. And then what will happen is when you program this thermocycler, okay, to move, how do you say, the change between these 3 different temperatures, you then program this thermocycler, the cycle part, to go through these 3 temperature changes over and over and over again in a cycle. Normally, you would ask the thermocycler to go through the 3 temperature changes 25 to 40 times. Now, why do all that? Because each time the thermocycler goes through those 3 temperature changes, your molecule of DNA, that DNA you're interested in, will be replicated. And then when it goes through that 3 temperature cycle a second time, those 2 molecules will be replicated into 4. And then when you tell that instrument go through those 3 temperature changes again, your 4 molecules of DNA are replicated into 8. So what happened in this especially advanced forensic science is that what scientists that were not familiar with the technique but became familiar with it came to realize is all I need to do is pick up, isolate, or pick up or isolate one molecule of double-stranded DNA. And then if I take that one molecule and use it in PCR, I can increase the number of copies of this one molecule into the billions, into the millions or billions. Now I create such a high concentration of this one molecule, it's easier to analyze. So, purpose of PCR, DNA analysis such as DNA fingerprinting for paternity testing, but DNA fingerprinting is also used for forensic science. So you might see somebody at a crime scene with a long Q-tip swab, okay, has that little cotton end on one end, and you'll see them going around wiping surfaces in a crime scene. Yet when you look at them wiping the surface, you don't see anything. There's nothing there. Well, what they know is that there might be one molecule of DNA from one skin cell, say, that nobody can see. But if they can pick up that just one molecule from that crime scene, from the person that committed the crime, if they can just pick up one molecule, they can use this technique to turn it into millions of billions, making it easier to analyze the DNA and thus figure out who the suspect is. Well, between all these techniques, DNA cloning, gel electrophoresis, PCR, okay, we were able to create GMOs, such as GMOs that produce human insulin. By the early 1980s, a company by the name of Genentech was credited with discovering and isolating the human insulin gene. They not only discovered and isolated the human insulin gene, they put it in cloning vectors to make copies of it. Eventually, they didn't have to use these vectors. Eventually, they were able to use PCR. But in the early days, in the early 1980s, they used cloning vectors to make copies of the human insulin gene. Then, as an added bonus, they realized when they took the cloning vector containing the human insulin gene and used it to create GMO bacteria, the bacteria would express the human insulin gene, resulting in the production of insulin. And then what they would do is they would take these transformed bacteriums and they would grow them up in these large vats of solutions, supporting not only the growth of the bacterium, but supporting the bacterium so they would express human insulin. And these transformed bacterium would produce large quantities or concentrations of human insulin. And then what they're not showing you is that these transformed bacteria that are producing human insulin, they would then take this entire solution, put it through a purification process, eventually isolating the human insulin, and then marketing it, offering it to those that say we're diabetic, such as type 1. Prior to this company, Genentech, okay, coming up with this innovation through DNA technology, type 1 diabetics in particular were being given pig insulin because pig insulin was the closest in homology to human insulin. The problem was it wasn't 100% identical, so they were having some serious side effects. So this was a huge advancement using these different techniques, everybody. As I said, PCR and DNA gel electrophoresis can be used for DNA fingerprinting. DNA fingerprinting can be used for paternity testing, even maternity testing, or forensic science. The first use of DNA fingerprinting proved one man innocent and another man guilty of murder. So this was huge. They used to use blood type, and that was horrible. It was found to be absolutely horrible. You cannot use blood type. You had that pattern of inheritance bonus lab. And if you did it, the last Punnett squares you did had to do with blood type. It's possible for dad to have blood type B, mom to have blood type A, and you have type AB or type O. So it was horrible to use that. But as technology advanced, one thing I don't mention here is DNA sequencing advanced tremendously. So that by the 90s, they had sequenced enough of the human genome and analyzed the human genome to realize most of human DNA that forms your 23 pairs of chromosomes, it doesn't code for anything. Only 1.5% codes for genetic information. However, in what the rest of that DNA, which they used to call junk DNA, they found out it's anything but junk. It contains tandem repeats of different kinds. As we talked about before, they're showing you 2 examples of tandem repeats. A tandem repeat could be made up of 4 bases, AGAT, that repeats over and over again along a single stretch of DNA, side by side. Or there are other types of tandem repeats that could be GATA that repeat over and over again. But these tandem repeats, what they found out, okay, is that there are short ones and there are long ones. And you can find them on basically primarily the first 22 pairs of chromosomes. And what they found is if they not only analyze these tandem repeats, but they analyze the different types located on different chromosomes, they can actually link somebody to a crime scene, or they can identify who is the biological mother or father through DNA fingerprinting by identifying what type of tandem repeats, okay, do we find in this person that we find in the DNA from this other person or from this crime scene. And this is what they're showing you here. And then what they'll do is they'll use PCR to amplify these tandem repeats. And they already know, they already have an idea, okay, of the size of each tandem repeat. Therefore, if a person inherits, a tandem repeat that repeats 8 times, they already have an idea how large of a piece of DNA that would form. Therefore, when they perform agarose gel electrophoresis, they already have an idea, okay? When they compare to the crime scene, they can actually equate the number of times that repeat occurs to the size of DNA that would form. And then once they compare the two crime suspects' DNA to the crime scene DNA, they just look which of these is 100% matched. You can see suspect 2, the 5 sizes of DNA corresponding to the different number of tandem repeats matches up perfectly with the crime scene. If you look at suspect 1, 4 of the 5 fragments match up with the crime scene. However, one of them doesn't. All it takes is one mismatch, and that exonerates or proves that this suspect is innocent. Now, when they started introducing DNA fingerprinting, okay, in forensic science, in court, well, the people in court, they didn't know a damn thing about biology. So they weren't accepting it in the beginning. It took time. But eventually, the court system, and lawyers and judges and lawmakers came to realize, whoa, wait a minute. This is great. We need to use this type of technique, DNA fingerprinting. Okay, because we're almost near, how do you say? We can 99.9% identify who the person is. Okay, who the criminal is, or who the mother or father is. So here, this is just showing you what I showed you before. If it's DNA from the crime scene, they'll identify different types of tandem repeats on different pairs of homologous chromosomes. And then the suspects that they obtain DNA samples from, they will analyze the same tandem repeats on the same pairs of chromosomes, and then they'll just do a comparison. And again, as I said, they already know from the crime scene how many times the repeats occur on each pair of chromosomes, and each repeat corresponds to a certain size fragment. So if the repeats happen up to 11 times, in this case, it's going to result in a large piece of DNA. But if for any of these repeats they only happen a couple of times, that lets the scientist know, hey, I'm going to end up with a small piece, and then they just do a comparison to match up. Okay, I know it's a lot of technical information, but use your notes, feed off of your notes primarily, okay, for chapter 12. I know I said a lot of extra stuff, but feed off of your notes for the test, okay? The test is still a take-home test, but I moved it up. So please notice on Blackboard announcement, I moved your test up to Tuesday. I believe it's the 12th, if I recall correctly. Let me double check my phone here. Sorry, my days are all mixed up right now. Okay, so I moved your exam up to Tuesday, May 12th. You'll have 48 hours to complete the test and submit it to me, okay, by 8 a.m. on Thursday, everybody. It's the same as before. Nothing's changed, okay? As far as your grades go, I'm trying to get everything graded, but we're past the drop date, everybody. Okay, but I will try to get you your grades, but we're past the drop date. So you've got to complete the semester. Please make sure that you dig up that email about your options for the semester, because we did have students, we had at least a handful that did not improve from their first to the second exam, yet they hung around. So, and that's fine as long as you accept responsibility for your final grade, okay? For those of you that did improve some from the first or second exam, but you still didn't feel it was high enough, well, You now have an opportunity, not just with the third exam, but the fourth exam. Put forth a lot of effort. Take advantage of the fact it's a take-home test to improve your chances of doing well and being successful in the course, okay? Are there any questions? I'm going to stop the recording.